Thanks. Okay, thanks, Diana. Thanks to Ben, Diana, and Michelle for putting in a lot of work into this conference. It's a treat to get together and be able to discuss some things about what's going on in the center. And it's nice of them to welcome me to the center. I'm kind of new to the center, just joined last year. I guess everyone's kind of new because the center's just beginning. I'm going to talk about listening to the nervous system, and I'm going to try to create a metaphor of sound, which was quite nicely introduced to us by Ben. And I might mess it up because it's the first time I've ever tried to do this. But I should first start with an origin story. Everyone kind of has an origin story of where they come from. And where you come from often helps identify the way that you describe things or that what your interests and hobbies are, which can give information about how you think about something. And so when I was in high school, I guess I was in junior high when the origin story really started. But my mom got some sickness, ultimately passed away, and I got really interested in blowing off steam by lifting weights and started competing in powerlifting. And I was really interested in how my body would get stronger, my muscles would get stronger, and the principles and the things I had to do to make my bones and muscles stronger and be able to lift heavier weights became interesting. And so I decided I wanted to go study kinesiology, which at the time to me was a big buzzword and it was really cool. So I just wanted to study kinesiology. So I'm gonna skip a bunch of steps in this story, just in the interest of time. But now I'm kind of to a point where I'm starting my own lab. Apparently this clicker stopped working. Ah, there we go. So now I started my own lab with a mission. Set it away. Started my own lab with a personal mission in mind. And so when we were kind of doing a lot of these talks about our own labs in the kind of the last 3 years, we had to create ways that we could visualize things on a screen. And so my visualization came from my time in Chicago. And so Michael Jordan is here. He has a ball in his right hand, and he has an empty hand in his left hand. So the area that's holding the ball, we consider that high performance. He's able to hold the ball, he shoot the ball, do layups. Empty hand is not able to do much, so we call that impairment. And basically with injury, we can slide over towards that empty hand or we can move over towards the hand with the ball. And the goal of my research is to understand basic mechanisms within the body to slide people from that empty hand to the full hand, or understand why we slide towards the empty hand. So to do all these things, I really need to understand how humans actually control movement. And so this is a very simplistic design, but we have some goal in mind that comes from our brain, and we send some command signals down our cortical spinal or reticulospinal pathways. And there's eventually some kind of communication with motor neurons in the spinal cord that sends signals out to the muscle. Those muscles contract. That's the bare bones of it. What we don't often think about is that there's not just one motor neuron and one muscle that gets activated. And so there's interactions between all these neurons in the spinal cord that create really intricate movements. So we can move our finger this much, or we can move our finger this much. And it's not just one neuron that does all that, and our brain says, okay, neuron, do more, neuron, do less. There's lots of them, and they need to interact with one another. And so this is where the audio system comes in. So we have a mixture of signals that could be an audio generator or it could be an instrument or whatever the case may be. That's coming from the brain and the spinal cord. Then we have the motor neuron. That's our amplifier. That's going to either turn signals up or turn signals down. And we could have multiple amplifiers together. And then we have the muscle fibers. Those muscle fibers are then our speakers. And so they're like a cymbal. When you hit a drum, if you hit one cymbal, you get one sound. If you hit them repeatedly, the sound grows. And so that's how we actually control force. The more times we hit the cymbal, or the more times that neuron fires sequentially, creates twitches in the muscle. And those twitches start to fuse together and forces grow. And so that's represented on the bottom of the screen here. So on the left, it's hitting that cymbal really slowly. In the middle, you start to hit it a little bit quicker. Over on the far right, you start to hit it even faster and the sound grows or the force grows. So how do we actually record the sounds of our movement? The technique that I use and many others who will be speaking today use is something called electromyography, or the electrical activity of the muscle. And so traditionally, we would stick a needle into the muscle right next to some fibers, and that would be like sticking a microphone right next to my face, and you can hear the sound really well. Other people, if I go over here, put their EMG electrodes on the surface of the skin. And so they're further from the source, and it's really difficult to understand what that individual motor neuron is saying to us. So here, needle close to motor neuron or muscle fibers, good information about motor neuron. Put that electrode far away, tough to hear what it's saying. But I did mention needles. And so I'm trying to be hip with the times and using these AI image generators. So I typed in, man with needle, scary. And this is what it gave me. So I don't like needles. I'll use them if I put them in my body if I need to, but I don't really like them. I don't want to just stick needles in my muscles everywhere to try to understand what individual motor neurons are telling me about my movements. Another problem with these approaches is exactly what Ben identified as difficulties in hearing when there's multiple sounds. So I said there's multiple motor neurons interacting together, and if they're all doing things that contribute to the movement, you might actually not hear what the one you're interested in is saying, and you might confuse it with another one. And that's identified in this cartoon if you read carefully. Another problem I mentioned. When I was over there, it was hard to hear the very specific details of my voice. And that's because the noises have to travel distances and go through space, which turns into something called a filter. So this filter takes out the really high frequency components of a signal that we're interested in, and it kind of makes everything within the muscle look the same when we look at it from the surface of the skin. That's a problem. We can't identify one from the other. So a solution that many people, a lot smarter than me, have come up with is this idea of putting many different sensors onto a muscle and treating it like a cocktail party or an audio problem. The cocktail party problem, many undergraduate engineering students have to solve this one, is that if you put multiple microphones around a room at various distances from the people who are speaking, you can start to triangulate that sound. We try to do the same thing with electromyography, and this gives us the neural code for human movement. So now that we have these motor neurons and we have lots of them working together, what can we actually learn about humans, health, and performance? So the basics of all of those commands I told you from the brain down to the muscle are comprised of three main signals. So we have excitation, that's the go. We have the inhibition, that's the no. And then you have neuromodulation, which is going to modify how much that go and how much that no is effective. And so if we look at a cat motor neuron and we just increase the amount of excitation, so now we're just talking about the red signal, and it's represented by this black triangle on the screen. And we increase, so this is excitation telling the motor neuron to go. And then on the y-axis will be how fast that neuron fires. So if we don't have any monoamines, it kind of tracks, the pink line, it kind of tracks the excitation of that line. If we put a bunch of monoamines onto that neuron, what happens is it accelerates and fires much faster and continues to fire much longer with the exact same input. And this is this idea of neuromodulation. So I'm really interested in neuromodulation and how it affects motor neurons in humans. There's vast differences in the way that motor neurons fire in humans when you go from muscle to muscle. And we think this has really big implications for the tasks that we do with individual muscles. So I'll point out a couple things on this slide here. Can you see the mouse? On the left here, we have a shoulder muscles. These are the ones up here. Those muscles are really important for stabilizing our shoulder when we want to do a task. And then we use our hand as an endpoint effector to either point at things or manipulate objects. But at the end of the day, these muscles need to turn on and pretty much just stabilize us. And they don't want to, do these intricate behaviors as much. And so what you'll notice is that these ones have this steep bit on the front. it doesn't really change its discharge rate, and then it has these long tails that come down, very much like those cats with lots of monoamines on the neurons. In the hand, on the other hand, we have neurons that look like low monoamines. So they go linear up, linear down to the same behavior. So both of these behaviors are increased force, decreased force, just like that current injection I showed you on the previous slide. So what you notice is that different muscles either receive different inputs, which is quite possible, or they have different levels of neuromodulation, which could have really important considerations when we're trying to understand how these things work in disease, which I'll talk about in a moment. We can also look at things like aging. We all age, hopefully. And so this is work by Altamash Hassan in Chicago. And what we did here is we put these arrays onto human muscles, biceps and triceps muscles of the upper limb, and we had people produce these torque ramps. So a lot of these slides, you'll notice they just produce some kind of force. They increase their force, they decrease their force. And what you notice is that young people, well, we tend to have these patterns that look like those cat motor neurons, and they have this behavior that would indicate neuromodulation. One of the things that happens with aging is something called inflammaging. So you get a lot of inflammation, and that affects monoamines in the nervous system. Turns out that older folks, well, they have more of these symmetrical patterns, which indicate that they may have a reduced monoamines. So we can start to target things that might, or try to understand why aging affects motor control now that we know that the motor neurons fire in a different way. We also look at something like stroke. So hemiparetic stroke affects the brain. Those signals can't come down to those motor neurons properly. And to compensate for that, our brainstem tends to send many descending monoamines to the spinal cord. But we've never really been able to quantify that well. So here we have control participants. This is work by Altamash again. Here we have the non-paretic side, or the less affected side of an individual with a stroke. And here we have the paretic limb. And what you notice is that the way those motor neurons fire is very similar between a control participant, someone who has a stroke but is the less affected side, versus their more affected side. Again, an over-exaggeration of those monoamines. They're unable to modulate the discharge of those motor neurons, which then could have implications for their actual motor control. What about the effects of biological sex? So many of these studies have been done, typically in males. And we had a PhD student, Sophie Jens, come to the lab, and she was really interested in quantifying how these things differ between males and females. So again, similar techniques, triangular ramps, and she found that these estimates of the monoamines were much greater in females. Okay, so what does that mean for aging? So now she's working on a project where we're actually measuring sex hormones, things like estrogen, progesterone, and correlating whether those levels of hormones, which fluctuate throughout the menstrual cycle, actually then affect these estimates of the monoamines. Thing about aging in females, many of us know, is that post-menopause, there is a reduction in those hormones. And there's a lot of negative consequences post-menopause for females due to the reduction in those hormones, we think. And so if those hormones are facilitating these monoamines and then we have a big drop-off, that might be a contributor to why aging affects females more than males. We can also look at how interventions can be used and understanding interventions in things like spinal cord injury. So one of the techniques that I've been using over the past few years is something called acute intermittent hypoxia. This is basically breathing in low oxygen air for 30 seconds to a minute, and then giving someone room air for another minute thereafter. You repeat that sequence just like you were doing sprints on a bicycle. This is a map of EMG, which I didn't really show you on the bottom, but this is the force that someone with a chronic incomplete spinal cord injury was able to produce before, immediately after, and 60 minutes after this intervention. Turns out, if you go back into the 1980s, someone stimulated a carotid body chemoreceptor, and that causes the release of serotonin. Serotonin is one of the brainstem monoamines that I've been talking about all along here that facilitates motor neuron function. So what we did is we used these techniques, got someone to do a maximal contraction, which is here, pre-intervention, gave them this breathing technique, and looked at the discharge rate of their motor neurons. Just like hitting that tambourine faster and faster, making the noise louder, these people were able to fire those motor neurons faster, create more force, and improve their function. So we're able to start to understand why this stuff can have profound effects on human health. So what we found so far is that motor neuron properties differ across the muscles. Aging affects them. Neurological impairment affects them. Motor neuron properties differ between the sexes. And that we can use these motor neuron properties to start understanding therapeutic interventions such as acute intermittent hypoxia. It's all fun and dandy, seems exciting, but all of these are done without any movement. So future work with some collaborators, some of which are here in the room, is to use these techniques in a situation where the muscles actually perform movement. Because we don't very often sit down at a desk and pull against the desk to understand how we move. We don't do that very often. So the goal is to understand how we walk, how we actually move our limbs, and how the motor neurons function in those situations. So these are some people that were involved, picture from Banff at a conference. And with that, I have the privilege of introducing Dr. Dwayne Button. He's the Associate Dean over in HKR and professor. He will talk about intense exercise and whether it's a friend or foe. And interestingly enough, Dwayne to me is both a friend and a foe. It depends on the situation.